You are looking at a string of raw genetic code. Isolated, it is completely meaningless, just a scattered collection of biochemical letters without context. In this video, we will take that raw sequence, format it correctly, run it through the National Center for Biotechnology Information Database, and pinpoint exactly what organism or function it belongs to. Step 1 is simple. Open the file containing your raw sequence and copy the text. Before we move to the tool, look at the letters you just copied. You need to verify if the code is made of nucleic acids, DNA and RNA, or if it is a chain of amino acids representing a protein. Biological databases are strictly separated by molecular type. Identifying whether you hold a nucleotide or a protein is the mandatory first step to prevent an immediate search failure. Navigate to the NCBI BLAST homepage. You will immediately see that there is no single search button, but rather a set of distinct algorithms to choose from. This decision tree diagram shows how to route your data. If you have a nucleotide query, follow the path to the BLAST-N program. If you are starting with a protein sequence, you will route that input to the BLAST-P program. Warning tone. If you select the wrong program for your sequence type, the system cannot translate the inputs. The result will be a guaranteed error. To ensure the algorithm can read your input, we use a universal standard called the FASTA format. This is a mock interface displaying a properly formatted sequence. The first structural rule requires a single-line description header, starting with a greater than symbol, followed immediately by a hard line break. The second rule governs the raw data. The biological sequence must sit on the lines directly beneath that description header, completely free of any extra spaces, numbers, or special characters. With your text structured correctly, copy the entire block and paste it directly into the main query box on the BLAST interface. Following these strict formatting rules tells the algorithm exactly where your human notes end and the searchable biological data begins. Scroll down the interface to the section labeled Choose Search Set. Here, you must select a targeted database. If you suspect your sequence is from a human, select the human genome. For broader searches, use the standard NCBI nucleotide collection. There are advanced options below this to filter out low-complexity regions, but as a beginner, leave these on their default settings. At the bottom of the page, click the primary BLAST search button to initiate the run. The page will automatically refresh to a loading screen. Depending on server traffic and query size, you will typically wait a few minutes for the results. While you wait, the algorithm is taking your single query and performing millions of statistical comparisons against a global library of documented genetics. Once the processing is complete, the results dashboard appears. This dashboard organizes the data into 3 distinct sections. The first section is the graphic summary. It provides a high-level visual heat map showing where your query aligns with known sequences in the database. Scroll past the heat map to the description section. This is a ranked list of all the biological sequences that match your input. This abstracted data table represents that BLAST results list. The most critical metric to look for is located here, in the column labeled E-value, or expected value. The E-value operates on inverse logic. As values drop below the 0.01 line, the likelihood that the match occurred by chance decreases, making the biological significance of that match much higher. An E-value of 0.01 or less is the success threshold. When a result reaches exactly 0.00, you have mathematical proof that your match is biologically real. Isolating the E-value allows you to discard the random mathematical noise and focus entirely on proven biological evidence. You started with a raw, disconnected string of letters. Now you have a verified, identifiable gene or protein sequence with established context. You extracted a single, verified biological identity from the largest genetic database on the planet. With this verified match, you can trace evolutionary lineages, pinpoint specific mutations linked to diseases, or identify protein structures required for pharmaceutical design. As your research scales up to handling massive, multi-sequence datasets, you will eventually transition from this web interface to using the BLAST Plus command line tools for faster processing. Every major medical and biological investigation relies on comparative genetics. Formatting a sequence, selecting the right database, and verifying the mathematical significance of the match is the foundational skill required to conduct that research.